we have a block of rooms booked out for those that are deciding to participate. Okay, so I'm gonna stop that, and then I'm gonna really introduce our speaker today. I'm super excited to introduce Rebecca Taylor, who is joining us today from CMU. And so Rebecca is the ANSYS Career Development Associate Professor of Mechanical Engineering and BME in Electrical and Computer Engineering at CMU, as I indicated. And what's really neat about Rebecca is she's got a very diverse background and very diverse training. And so she did her undergraduate engineering degree at Princeton in mechanical engineering. And then from that point on, she moved to the West Coast to do her PhD at Stanford. And she did this with Beth Pruitt. And then she loved Stanford and decided to continue on a postdoc there. And there she did work with James Spudich, who is in the Department of Biochemistry at Stanford, and someone who is really a pioneer in studying motor proteins and conducting some really amazing single molecule and force spectroscopy work in that area. And then in 2016, she joined the faculty at CMU, and in her lab, you see that same diversity of training coming out in the work she's done. And so it spans from mechanobiology, as we will hear about today, to robotics and some manufacturing applications and fabrication, top-down fabrication, bottom-up fabrication. And for her work thus far, she's received a number of accolades and awards, the NSF Career Award, the Air Force Young Investigator Program Award, and then the CMU Dean's Early Career Fellowship. And so she's really won a lot of awards. Super excited. I met Rebecca actually in person at a Gordon conference in Southern California a couple of years ago. And it was really a pleasure to meet her, and I'm super excited to hear her talk today. So Rebecca, the floor is yours, and I see your slides.
Fantastic. Thank you so much, Khalid, for the really kind introduction. I'm so delighted to speak with you all today. I have the chat open, like I can actually see it. So if you I don't know, if you have questions that you want to ask throughout, I can actually pause and address things. We could also, I guess, answer questions at the end. But yeah, so here I am. We really work across fields, and I'm so thrilled that the field of mechanotechnology, is growing. I think so much. And the idea for much of our work is basically getting a handle on how do we communicate with cells? How do we make visible those forces that occur at the micro and nano scales? And then also increasingly, how are we going to interface with and manipulate those environments? So like thinking about robotics and manufacturing. And so those are the sorts of topics that our lab explores. And that means the structure of my talk is kind of going to be 4 stories. There's 2 bigger stories and 2 smaller stories. I'll start with talking about specific probes and platforms we've made for mechanobiology and bioengineering. And then I'm going to move to talk more about actuators, these micro swimming robots in particular that we've been developing. And then I'll sort of wrap up by talking about how we think we need to push this field forward. And the 2 topics there are advancing automated design to really further explore the design space for these nanostructures, and then exploring interesting materials that give us novel properties for our nano machines. Cool. Okay. And then so what I always pitch to everyone, and I love audiences where this isn't going to be a hard sell, but nucleic acids are engineering materials, right? So they're great engineering materials for synthetic biological systems. If you believe that, it's a really important material for engineering. And in order to sort of bring in that diverse audience, to get people from multiple fields engaging with these structures, we actually make 3D printed models that are articulated and help people put together these structures. Also, as this is how I would remind people, once DNA is hybridized, it really acts like a hard plastic. So if you're thinking about how to think about it, uh, it's basically scaffolding that can be dynamic, but yeah, really useful material. Okay, so to get started, I'm going to jump into the first story about cellular interfaces. And a lot of this has to do with new ways to attach to cells based on differing criteria. And then also we'll talk about how to manipulate cells. Okay. So, so for this audience, you know, this is our motivation, and this may be many of yours as well. It's that with self-assembly based platforms, particularly DNA nanotechnology, uh, we can now self-assemble structures that are rivaling the complexity of the machines built by living cells, you know? And so if you want to make a synthetic pore, if you want to make a rotor that's actually driven by electrochemical gradients, if you want to apply, you know, piconewton level forces or communicate information across hundreds of nanometers, you know, and hi, Yanggong. These platforms can be embodied by DNA nanotech, right? One interesting example of this is when you think about a lipid membrane, you can engineer a device to attach to a membrane, to attach through a membrane, to make its way across the membrane, or to go inside an endosomal particle, right? So you, by virtue of engineering anisotropic nanoparticles with different functionalization, we potentially could learn the handshake, or the secret handshake, I would say, for how to communicate with cells. Okay. And we've spent a lot of time in the early years in our lab uh, demonstrating how to engineer these systems to interface with cells as we'd expect or as we'd hope, right? And so the simplest starting point is to say, when folks tend to think about a nanoparticle, you'll think about a sphere. And in that case, a multivalent sphere, for each additional binding site, you could imagine you're sort of they each have an independent binding probability. And so you can model this if you have more overhangs that can bind with a surface that's been seeded with complementary overhangs, you can get the behavior you'd expect with multivalent nanoparticles. It's fantastic. It's wonderful. With DNA nanotech, it is no small feature that you can have, you know, precisely kind of stoichiometrically how many binding sites you want. Okay. But things get even more interesting when you start playing with anisotropic structures. And so here we have basically a sheet of paper. You know, you could build anything, and we built a rectangle, and I know that that's somewhat disappointing, but it's useful here to illustrate a point. And it's that we don't see the same sort of thing we saw with our sphere. We can just increase the number of overhangs, and we don't really get more binding. And that's really disappointing. And the field had known this. And in fact, you needed a tether in order to get these structures to attach, right? And so one of the things that we investigated to and based on early papers saying that corner attack was likely helpful, if you put those overhangs at the edges, you see a robust restoration of that binding ability. So here we have the same nanoparticle, and depending on how we decorate it, we can get it to go bind with a cell, even in the absence of a tether. And this speaks to a really powerful potential for selectivity, right? Because we can basically, we can make a particle good or bad at its job based on how we decorate it, right? Um, so how would you use this? Okay, as a probe, let's use the bad one. So say we have a bad rectangle. We've decorated its face. You need a tether. And so just to demonstrate this, basically we use this bridge of differing lengths, and we can show that the longer the bridge is, the more attachment we get with the cell, okay? Now, without a bridge, you don't bind. But if you do something that hurts that protective glycocalyx layer, if you use trypsin or if you use neuraminidase, and you basically digest away or remove some of the charged elements of the glycocalyx, you can then actually restore binding without the bridge. So in this case, because the nanoparticle doesn't just bind well to everything, we could actually engineer it to help us find cells with a damaged glycocalyx. So we envision this as sort of a vascular paint type technology where you could in real time assess the functional intactness of some cellular protective structures. Right. Okay. All right. So here's another application. So one of the things, speaking about piconewton and nanonewton forces, uh, if we want to study human blood platelets, these cells will activate if you disturb them in the least, and that's their role basically. They're supposed to patch a tear in a vessel, right? So they sense mechanical disruption, and then I think of them like a folded up tent, like the ones that fold into a circle, and then you just sort of toss it in the air and it does its thing. Um, you have to be really delicate with them. And so it's actually been really hard for researchers to understand how to study the process of mechanoactivation, simply because they're so sensitive. And so we use the principle of binding with highly effective edge decorated rectangles to demonstrate that we can actually attach one of these DNA origami platforms to human blood platelets without activating them. And so here's the data that you would see where basically there is an optimal decoration density. And if we decorate with 6 edge decorations on our rectangle, we can get the brightest decoration, the highest decoration. And we can actually, because they bind so effectively, do this in a way where all of the conditions don't lead to p-selectin exposure, which indicates mechanical activation. They don't cause a significant change in activation, right? So we've got this platform, and now if you start thinking about it as a lab on a chip, there is broad potential for these nanoscale lab on a chip platforms that could do sensing, right? That could perform, um, yeah, chemical or mechanical assay to we can attach them to even the most sensitive cells. And so I like the superhero analogies and saying instead of getting the Hulk, this can keep this thing in a Bruce Banner kind of state. Okay, so here's the third, our third probe study. And essentially, I wanted to give you a summary of how we can attach to the membrane of a cell. All right. So in the first case, there are scavenger receptors. So sort of in non-targeted, even with non-targeted DNA origami, you can get some level of attachment and uptake. What we typically would do is what we call cholesterol anchoring in a 2-step manner, where we introduce these anchors that are cholesterol modified and they embed. The cholesterol embeds in the hydrophobic core of the membrane. And then you've got this single-stranded DNA sort of decorating the surface of the cell. If we attach the complement to our origami, we can get them to land and attach. Uh, we've also shown that you can actually feed azide sugar to cultured cells, and they'll actually build that block into the glycocalyx, which we can then click onto to attach the anchor DNA for our origami. So we can effectively choose whether we attach to the trees or to the ground beneath the trees. And then third, out of Ulrich Kaiser's lab, and I think I have this on the next slide, there are methods for doing this in one step, where basically if you sterically shield your cholesterol, you can actually put lots of cholesterols on an origami and not cause it to aggregate. And that way you could potentially have an in vivo compatible method to send these nanostructures to interact with cells. Yeah, okay. So what do we do with this? So I want to talk about why anisotropic nanoparticles are so incredibly useful. So just to start, spheres go in the best, then rods, then tiles, if they are, or attach the best, if they don't have any specific decoration, right? What's interesting is that internalization seems to be roughly proportional to how much attachment we get at the surface. Okay. But what we then try to show with decoration of these anisotropic nanoparticles is what are the benefits of decoration? And so for the sphere, it's already so good at going into cells that as you add overhangs, indeed, you get more and more and more attachment and uptake. But the ratio it's far less improvement relative to the starting point. Whereas with this tile, as we showed in the previous study, as few as 4 overhangs at the edges performs much better, much better than one that's face decorated densely, and then far better than one that's undecorated, right? So we basically can turn on the functionality of these nanoparticles, and that can be really useful if that attachment moiety allows you to, say, target a certain cell type. Because then you basically know signal to noise wise, you're getting attachment to the way you want it, not from untargeted means. And then likewise, it still holds that it seems like uptake is proportional. And I didn't show this for this paper, but we also sort of interrogated the different pathways. And one thing that we thought was interesting was even for our click chemistry method, where we attach to the top of the glycocalyx, they all utilize multiple endocytic pathways. Yeah, but there was really great similarity between how this works. So somehow they're all triggering uptake via clathrin and caveolin mediated pathways. Okay, so that's a lot. But here I also just want to say one thing that's really exciting is building on this work from Ulrich Kaiser's group is we then did head-to-head comparison for 1-step. And so the idea is even outside of a well-controlled, dish experiment with cultured cells, one day we could imagine having cholesterol decorated origami be delivered in vivo because you could just deliver it to the blood and then it could like attach to the cells. And so the fact that the light green is more than half the height of the dark green is fantastic news for us. Yeah, so there's multiple ways to do this, and the takeaway is that you can actually then tune your uptake as well. And now this is particularly interesting because what we find is that through confocal imaging, these particles can get to the nucleus just using this approach, right? And so ongoing work in the lab, I would say, is we're trying to understand how to leverage this. But if you can get to the nucleus, I think suddenly you also have the opportunity to bring in gene cargo. Okay, nice. Okay, so this is my 4th story of this, or of the first part of the talk. And this one's basically to say, okay, so we can be really delicate. We can send the nanoparticles to exactly the cell type that we want. It's super, super, I don't know, precise and careful. Well, why don't we actually try the opposite and try something very drastic? And so in this case, we decided to see if we could construct a synthetic cytoskeleton on the outside of these cells. So we use our click approach where we anchor to the top of the trees. In this case, the glycocalyx is shown in purple. And we're basically anchoring these long 6-helix bundle DNA origamis to the glycocalyx and then selectively cross-linking them. And we can tune the number of cross-linking sites between what we call rod A and rod B. Okay. So in this case, my superhero analogy is that now we're trying to make cells into Iron Man. And if we do that, the first takeaway is indeed, when you have rod A and rod B, they're very nice, they don't aggregate. But when you mix them, you do get a sort of gel-like system. And that they can also remain without degradation for up to 30 hours in media, even containing serum, which is exciting. Okay, so what does it do? Okay, so the first thing is, how does it mechanically alter the cells? So we did micropipette aspiration studies and actually showed that you could triple the effective elastic modulus of the cell. And you could actually tune how much you modulated this elastic modulus by tuning the number of overhangs that interact between rod A and rod B. And interestingly, even just having rod A did actually appear to make an improvement or an increase in elastic modulus. So we thought that was really promising because it's so tunable. But then we wanted to see if it had an actual, what other biophysical impacts it could have. So we looked at the membrane mobility. And so using a FRAP assay, we could basically look and see what happens to the mobile fraction of lipids and how long does recovery take. And in this case, as we build this shell, we reduce the mobile fraction and we also increase that recovery halftime. Okay, so, and we can do it in sort of a graded manner. And finally, we wanted to show what is it useful for. And so here's where things get fun. Now, this is an assay that is not very nice to do with cells, but we put them in very low salt concentration solutions. And this osmotic swelling can lead them to rupture. And so what we tested was how does having just rod A and the entire nanoshell alter what happens to the cells? And we can show that basically the dark blue is the signal here showing you don't actually get a big change in cell size as you decrease from 0.9 to 0.3%. But most notably, they haven't ruptured, even in 0%, right? So as a temporary ruggedization tool, this could be really useful. And then you probably wonder, do the cells actually survive better? Does it boost survival? So we put them through a triathlon of challenges where we said, okay, we'll osmotically swell them, and then we'll check viability. We'll put them in centrifuges, and then we'll check viability, and then we'll just repeatedly inject them through a syringe, which is the kind of thing you would do for cell delivery. So it's actually really relevant. And in all cases, we had the rod A plus B condition lead to higher cell viability with substantial improvements just from rod A, right? So in terms of benefits of DNA origami, this is potentially really versatile and really powerful for temporary ruggedization of cells. Okay. All right. So that's the first part of my story, or the first of the stories. So let me completely switch gears. And like I said, I could pause and take a breath and say, does anybody have any questions here that I could answer? Which cells do you use? So we use Jurkat cells, not because we wanted to study the immune engineering, but because they were not adherent cells. We've also done this with HUVEC cells, and we're now doing it with HEX cells and also investigating some other cell lines. So they do attach on adherent cells, but I think it's easier to get a sense of the uniformity of the decoration. So we started with Jurkat. But otherwise, we have seen it that we can lift them off the surface and then coat them. So you could potentially get sort of a spherical coating on even adherent cells. Thanks. Thanks. Okay. And then how long does the benefit last? And we did not study a whole lot longer than a couple days. So do I have, oh, I don't have that slide. Okay, so I won't put that slide up. But essentially, that's one of our open questions. And what I would say, now that I've lost this, the questions that we're currently actually underway studying have to do with what happens to cellular metrics of health, over a period of multiple days. How can you maintain the performance of the shell? How can you think about changing the integrity of the shell to see if that can help it stay on the cell longer? So these are the open questions we have, which is basically, is it a 24-hour thing that you should then remove? Definitely. Our sense with most DNA origami is it's gonna start degrading immediately, but we're good at 30 hours. It might be good up until 72 hours, but we would assume some amount of degradation is going to happen without additional chemical protection. Yeah. And then, so yeah, oh, and that's another great question. So in terms of platelet activation, we also did TEM, right? And so we've got images of the sort of cell area, and we haven't seen structural reorganization, if that's what you're asking. And then I think the last one is, can cells still participate in cell-to-cell communication with the nanoshell on? So in our paper, one thing that we showed was that if we put rod A on one cell type and rod B on another cell type, and then we connected them, we could actually get them to create little florets. And so you can certainly have cells bind to each other, across this. I think the key feature that would be interesting to quantitatively characterize is how much communication can go on. because the origami are 400 nanometers long, you could imagine pore sizes would be hundreds of nanometers potentially in diameter. So I mean, or in, in effective size. So I think that there's the opportunity for a lot of communication to happen. And that's a fantastic question. And certainly, yes, like calcium signaling and cytoskeletal reorganization is a key part of this that I think is really interesting. And I would say we are likewise currently investigating. Cool. Okay. All right. So I'll change topics if we can return to any questions later. But I want to talk a little bit about, you know, looking at what's missing from reconstitution methods with nucleic acid nanotechnology. Um, we can see we can make filamentous structures. You know, we can potentially move material along those filamentous structures. And so it's really reasonable to imagine making transport systems that directly mimic what the cell does, right? But one thing that we could do that the cell doesn't do is pioneer new ways to move materials around that actively take advantage of external magnetic fields, or it could be, light or acoustic stimulation. But there are ways to get energy into systems to do work that we could leverage. And so I want to basically tell you this story about a tool for maybe getting your probe where you want it to go and using methods that the cell hadn't been able to leverage. And so here we talk about microswimmers. In its simplest way, if you had a little robot that could move on demand, you could use it for microsurgery, you could do targeted drug delivery, you could stir fluids, you could assemble chips, So I mean, there's a large potential there. And so we've been investigating how to make tiny swimmers that use the simplest possible components. So from a building perspective, what might those be? You could have DNA origami, you could have live cells, you could have colloidal particles of different types. Other building blocks could be biofilaments. So you could use, peptide sequences. You could use biofilaments like microtubules. You could also make DNA nanotubes. And so we picked the very simplest ones here. We've got polymer microspheres, and DNA nanotubes. And this is our robot. And prepare to think about a slow-moving robot. All right. So this robot's on the order of like 2 to 20 microns in size. And it's moving at the scale of microns per second. So it's very relatively pokey. But what I would argue is we can control how it swims, how it forms, and when it comes apart. And so this has the potential to give us everything we would need in a tool for moving material at the microscale. Okay, yeah. And then what we're most excited about is in order to have something like this actually swim, you have to overcome the scallop theorem. And the easy way to think about the scallop theorem is in low Reynolds environments, you really don't want to be a scallop because when a scallop rapidly closes its shells, it can push forward, but it doesn't have the benefit of having momentum really be a big component. Like inertial forces are very small relative to viscous forces at the nanoscale. So it doesn't just continue to shoot forward. Once it's done expelling liquid, it just closes. And then once it opens again, it actually pulls itself right back where it started. So if you were a scallop at the microscale, you'd have no way to move. If you are a rigid robot undergoing reciprocating motion at the microscale, you can't move. So you have to have a flexible linker. So this entire field of microswimmers is dependent on having this nanomaterial with a tunable flexibility. And DNA in this case gives us exactly what we're looking for. Okay. So then here's where we'd say, drawing on my experience in microelectromechanical systems and sort of more top-down manufacture, we used a templating method to bring these little polymer microspheres together and actually filter them so we get a higher uniformity than we otherwise would. And then we use DNA nanotubes to connect them. You can see this little collar here basically showing there's a fairly disorganized, but there's a connection between them, right? Okay. Anyway, so these are our little swimmers. I think they look like ducks. And then generally, when you put them in a constant magnitude magnetic field, that constant magnitude field doesn't apply a force um, through, like, you'd have to have a gradient in order to just pull, right? So what it does is it applies a torque. You can rotate a particle to align with the field. So if you then rotate the whole field, you can rotate the magnet back and forth and back and forth. And it has a really nice property where, like, multiple magnetic particles in solution under these stimulation conditions, they'll just sort of rock back and forth. They won't move. But once they link together, then you can swim away, right? So we've got a population where basically the assembly with the nanostructure can be read out based on whether or not that swimmer is now moving, right? Likewise, you can tell, is that linker intact? Because locomotion stops happening if they're no longer effectively connected. Okay. So these are our little swimmers, and we've had a lot of fun as mechanical engineers because one of the things that we realized early on was that heterogeneity in these structures is a nightmare for quantification. It's hard to ask how does changing the geometry affect the swimming properties. So we actually made some at the millimeter scale, which is enormous for us, right? But we used two-photon polymerization to make millimeter scale models of these, and then we could actually systematically investigate how doing things like changing the aspect ratio or changing the stiffness of that linker can actually change the frequency at which you achieve your peak velocity and also what the peak velocity is. Right? So basically, so now we have a tool that can act like our ground truth to understand how these particles should be designed and engineered at the microscale. We continue to push this forward because now we want lots of them. So one of our key advances recently was that Tara Imamura and my group realized that if we did a better job of tuning the surface energy of the template, we could promote bind or pocketing of our polystyrene particles. So basically, by raising the surface energy, she went from having a system that sometimes pocketed, or that pocketed enough that you got swimmers, but not many, to one that robustly can make hundreds, even on a relatively small template. So we could, it's like up to 30% filled, which is fantastic for us because then what that means is that you can then make combinations of small and large swimmers. And you can start making more complicated body plans. And because you got this higher yield, you can achieve even more complicated. And then even patterns with up to like 18 microspheres. So now we could say, okay, well, these swimmers can have arbitrary body plans. They can have flexible components. And we can really truly explore like how we should be building microswimmers. Oh, I also didn't say, we've shown that, making them responsive means you can easily take them apart on demand. Okay, so that's my second story, but the other two are really fast. So I don't know how much time I have left, Khalid.
Oh, if you had to end at 12:45, that would leave us with 10 or 15 minutes for questions.
Okay, okay. All right, well, so let me tell you what I really think the field needs. I would say that the field of DNA nanotechnology and sort of broader nano manufacturing of structures and machines, we need tools to help us understand how to explore the full design space. So tools for the generative design of these structures are potentially really powerful. So, well, here's an example that from my group where Tito Babatunde addressed the challenge that there wasn't an automated way to make arbitrary cross-section, multi-layer DNA origami, right? It's usually the kind of thing that you sculpt by hand. It's usually really time intensive. And so this tool basically encoded in a formal language what it takes to make honeycomb DNA origami. And then she can generate the scaffold routing for hundreds of possible particles. And she can do it based on an input desired shape, but also potentially on an input desired function, right? And we really want to move to approaches where we might say, give me structures with this stiffness, give me structures that deform this much, right? Or say things like, give me a structure and just use all the material I have to fill up a mold of a certain size, right? I want my nanoparticle to say be this tetrahedron, but when you're done using all the scaffold, let's just make it hollow, right? So she can actually direct how she fills it so that the user doesn't have to figure out how big can I make my tetrahedron ahead of time. That can just be solved, right? And then potentially, for example, different scaffold sizes could make nanoparticles of the same external dimensions. Okay, so it just gives more flexibility to the DNA origami world. Likewise, AJ Vetterini in my group, with work also in collaboration with John Kagan, has been working on advancing the automated design of wireframe DNA origami. And here, what we might want to say is for DNA origami wireframe structures, these are great for building lattices. They're great for accommodating nanoparticles. So a common work process might be that you know where you want to have nanoparticles, say in pink, so you could kind of exclude those areas from where the particle will be. And then you also might know in green how you want it to interact with other nanoparticles, or where you might want to have the attachment sites be if you wanted to, say, build a lattice, or just define the maximum dimensions. And then here we could do an optimization-driven process to figure out what shapes meet the desired properties. And that way, when you design a shape, you can do things like maximize porosity, right? You can also do things like sort of multi-objective design, right? So you can start playing with doing what we as designers do all the time, which is explore Pareto fronts that represent trade-offs between the different things we might want, right? Yeah, and so this is an example of just how these tools I think are really, really powerful and are going to save people a lot of time, but also finally let us formalize what we're missing in the design space. And then the last piece, which I want to go through, has to do with xenonucleic acids. And I would just say one of the key challenges when we think about DNA nanotechnology is that you better hope that degradation is a positive, because you're going to have it degrade, right? Like nucleases are going to break it down. Likewise, if you use it in the context of a system with an immune system, um, it may trigger inflammation, right? And so if we think about what a material might need to be in order to have superpowers, well, we'd want it to resist degradation. We'd probably want it to be high affinity, but we'd also want it to be able to bind to DNA, right? And so that's what PNA does. And here I thank Chris McComb for generating this DNA mimic cartoon for me so that I'd have a creature to point to. But yeah, but so PNA is really interesting. Its geometry differs from DNA quite a bit, but roughly you can think about it as being less tightly wound, right? So the framework for how to design with it can be simplified to be analogous to DNA. One thing we noticed was it wasn't something we could model with existing software. So here I want to do a pitch for the fact that we like making models. These are, things you can hold in your hands. And we actually build our motifs in PNA with our models to help us understand how should we actually be building with this unusual structural material. But yeah, so the DNA is in white backbone, and then the PNA is in purple here. And here's another pitch for something that maybe the only thing that isn't published yet is we have a paper out in the Biophysicist talking about this model where we engineered connectors that make it easy to make a nucleic acid DNA model, right? And so you can print these out and they're available, linked from the paper. But we've now also engineered the web-based CAD program Onshape. Is that playing? And we've made it so that any user can actually import a molecular model and then add the connectors automatically so that they can represent the different types of bonds and then have a kit that they can print out and manipulate in 3D. And so we think this is really important for both education, but also for just people working to understand the chemistry of their structures. So, okay, I think that's, oh yeah, the last part of the story. Essentially, we use that tool, and here is one example that we can build these nanofibers made with PNA. And this was one of the first works we did in PNA in the lab. And we basically just said, okay, this can undergo Watson-Crick-Franklin binding. like DNA. So let's just pretend it's DNA, but it's got 18 bases per turn. And so we designed a motif that should be able to form a 3-helix bundle. And then we made it. And first of all, without additional modifications, it's not very soluble in water. So we did most of these studies in 70% DMSO. We also did them in DMF. But when we built these things, they actually follow the program you'd expect from a DNA nanotube. We get multi-micron long filaments. They're fairly uniform, but we realize something is up here. And so when we zoom in, you can see that they're very clearly bundled. So I'd say that the sort of negative stain really highlights the light, dark, showing a bundled structure. So instead of it being like 6 to 8 nanometers across, we got filament widths up to like 80 nanometers. So they're tightly bundled. So it's actually kind of a material that's very, it can be very DNA-like and very peptide-like depending on the scale. Right. So really interesting engineering material. And we've been pushing forward how to manufacture it, how to do like isothermal formation. And we're also exploring the range of modifications to enhance water solubility. And I would say that would be sort of the next story out of my lab is what are we now building? But it has really interesting properties, like you can template its behavior by growing it off DNA, and so you can form long filaments if you have just a few templates and like short filaments if you sort of oversaturate with lots of templates. So we think it's a really powerful building material. And because it's not a substrate for nucleases or proteases, it changes how we can use these technologies and where we can use these technologies. Okay, so I'd say that's it. That's the sort of summary. And I appreciate you always staying with me. I know it's a lot. And I'm glad, Khalid, that you set them up to understand kind of the breadth of what I talk about. But hopefully today I've shown how these nanoscale manufacturing processes allow us to achieve complex cell manipulation and probe for things like membrane or glycocalyx intactness. We can also ruggedize cells, manufacture tiny robots, and that we're actively engaged in pushing these tools forward. I think the only other pitch I'd make is that we're also trying to figure out how to bring DNA nanotech to the broader community. So we've been investigating sort of automated methods for making DNA nanostructures. And so that work is out as well. And with that, I would just say that many thanks to my lab members, all of our collaborators, all of our funding sources, and to our facilities and administration. So yeah, thank you all so much.
Well, terrific. Yeah, we have a few minutes. We have time for questions. If, as Rebecca indicated, I think you can just type your questions in the chat, or you can unmute. And maybe I'll start off everybody with a question that was on my mind. It seemed this edge binding was very critical to get access to the cell membrane as opposed to on-face binding. And I was wondering, is this driven primarily because of electrostatic charge of the origami, or because it's highly negatively charged, or alternatively, is it a size, a steric barrier to binding? Do you have an idea of what the answer is there?
Um, so I think the simulations that folks have done of membrane interactions have shown this effect even in the absence of a complicated glycocalyx, right? So I think that charge is enough to explain it. You have a negatively charged membrane and a negatively charged origami. Unless you change the surface charge of the origami, it makes tons of sense that you'd approach on the edge. But then there just also becomes this other fun parameter of, well, it does have to navigate sort of the topography of the membrane as well, and how tightly clustered your receptors are could also play a role. So I think that the geometry of the origami is incredibly important, but you probably have both factors rightly.
I mean, and along those lines, so when you cross-link your with the rod A, rod B structures, and you change the mechanical properties of the cell, does that, I mean, you're using jerkouts and immune cells, does that alter their ability to bind and trigger and respond to antigen?
This is one of the questions we would like to ask. And so I don't know how to say this without directly telling you what we've currently proposed. But essentially, it's like you you can think about these shells as a backpack. So you could engineer them to have what the cell needs, but also to promote binding. Um, but in terms of putting the cell in a state of being stressed because suddenly it can't do certain sort of tasks that are essential for life. Yeah, I think we're going to see that there are limits to what we could do, and we have to figure out how to attach in potentially in places where it's not going to be as disruptive. I think that's one of the things I'm most curious about is figuring out what are the hot spots on the cell that we should be interacting with. Yeah, so I think that's a great question.
Great. You have a couple of questions in the chat. I don't know if you could see those.
Okay, so it says, how challenging is it to connect this technology with intracellular genetic pathway to make it more autonomous for cell therapy? Um, uh, the we are actively doing this, and so I'm really happy to have this question. And so yeah, so I think um, there's been so much exciting work from like the Castro and Dudna collaboration, as well as like the Dietz lab and Kretzmann work. And there are others. Sorry, I've just those two, but there have been really interesting work showing that if you fold up a gene and a promoter into a DNA origami nanoparticle, you can deliver that gene. So I think that the next natural question is, okay, so what do we need to know about nanoparticle design to get them there? And that question is right on. I think the answer kind of lies in uh, understanding shape, valency, and clustering, and then maybe the flexibility of your ligand decoration as well. Just to give the answer away. That's fun. Okay, and then I think so. Yonggang asks, what was the connector for the microbead rotor? And so this was we've been using Peng Yin's uh, work from 2008 to make the 10 helix nanotube. And it may just be that we were like, we tried a few different nanotubes, and this one had the properties we liked where they didn't aggregate or form networks as much. But what we do is we just form those tubes. And then the goal of that project was to be not sloppy, but to have a robust process that could be achieved even without careful control of the number of tubes. So we get many tubes, not just one. And I think, and the goal, and then originally, the thing that I think is interesting is originally I thought, oh, well, these connectors have to be more than a micron in length. So it's going to be hard to make origami that are that long. So these nanotubes fit the bill. But what we found is that these swimmers work really well even with shorter connectors. So we could actually systematically study them with origami. And then at present, you can tune the concentration of the tubes to change the number of connectors, but it's hard to be quantitative about it because the length of the connectors or the length of the tubes is so variable. However, if we start using origami, I think we can be highly quantitative. So I think that's a really fun direction.
So,
Uh, mhm.
Rebecca, maybe I won't type it in there. Just a follow-up question. I think you talk about that already for the motor. I think not only using origami, not only you can use it as a connector, but probably also equally interesting is that you can tune the behavior or reconfigure the system. So, for example, just like as you said, you could have for example, a rigid linker, a shorter linker, but then you can convert to a longer, more floppy linker that would change the motor behavior. Is something you have been considering to do?
Yes, 100%. Because I think on demand changing the mechanics of that connector would be a beautiful readout because then you change the swimming properties, right? So it's basically an assay readout, and all you're doing is measuring velocity of a particle. So I totally agree. I think the other thing that's cool is when we were doing this work, what we had found when we did our yellow submarine millimeter scale model is that the analytical models people use to predict how a microswimmer will move, those don't capture the roll-off that we see. And they're based purely on assuming that you have two particles and you're trying to calculate the time varying drag that those particles see. You don't take into account how they interact with each other. And so we see like the model doesn't work for higher frequencies of actuation. And so we actually now have a collaboration with ANSYS because they've got a sort of CFD solver that is really good at capturing both elastic material mechanics as well as fluid structure interactions. So we're working on a platform that will help us predict the swimming capabilities, even if those particles could be anisotropic. So I think coming back to it, the particles should be cells. They should also be big origami that have really interesting shapes. So I think that's another way to dynamically change swimming. Cool. This is so fun. Okay, so there's one more. So it said, could antibodies or stimulating factors be incorporated into the nanoshell to continuously stimulate the cell as the environment changes? I would just say, I guess I would say tune in. I will know probably in the next 6 months what we can say about that. But I think that would be the hope. Yeah.
Um, I see. I don't see any more questions, but I have one more last thing. You alluded to the fact that the structures get into the nucleus, and I was a little surprised because the nuclear pore complex has a cutoff, and some of these structures are certainly larger than what I think the cutoff is. Do you know why and how these DNA structures get into the nucleus?
No, I think that, we're going to have to do cryo ET at some point to really understand this. But if you flood enough of the particles into the cytoplasm, they end up in the cell. And it's not necessarily just the fluorescence. I can tell you that we've seen with gene encoding nanoparticles that you get expression. So one really useful thing to use DNA nanotechnology for is to design experiments where the particle asks the question for you. And so we have a few different shapes and we can compare how they do. It's not exactly mechanistic in terms of showing why they do what they do or what else might be going on. But we can definitely say like what works well for getting something into the nucleus. Yeah.
All right. Well, that was great, Rebecca. I don't see any more questions from either the chat or in person. And so I guess with this, let's all thank Rebecca one more time for a really wonderful seminar and participating in this series. I would also make a plug and say next month, Mingshu Yu is giving a talk, and so likewise you'll see similar topics of the interface of using nucleic acids to interrogate cells and mechanobiology. And with this, thank you again, Rebecca, and thank you everybody for attending.
Thank you so much.
Thanks.